Hello everyone, welcome to the ITLS Academy. Today we are going to discuss the molecular diagnosis, okay, or molecular laboratory. Okay, so what should we do? We use a molecular biological techniques uh, identify or to expand our knowledge about the natural history of the particular disease. And along with this, we have also find out the people who are very risk for that particular type of disease, okay, at their molecular level. The use of molecular biological technique to expand scientific knowledge of the natural history of diseases, identify people who are at for risk for acquiring specific diseases and diagnose human disease at the molecular level. So this is the definition of the molecular diagnostic. Now, molecular diagnostic is used to identify the to get a knowledge about a specific type of information in a sequence. Okay, so it is used to get a special type of knowledge, or you can say that a specific information. Specific information. Okay, so we take this information in the macromolecules. Okay, we take this information for about the macromolecules, and this information is used to identify the risk, okay, diagnosis, prognosis, prediction of response to therapy, and monitoring therapeutic responses. So these are what we use these technique for these things like your risk identification, diagnosis, prognosis, prediction of response to therapy, and monitoring therapeutic responses. Now come to the next slide. Macromolecules. Here, the macromolecules are what they are: peptides or proteins, polysaccharides, polynucleotides, or a nucleic acid. So this the interaction of this macromolecules like your peptides, protein, polysaccharide, polynucleotides, or nucleic acid to the molecule. Okay, so study about all these called a molecular biology. And even the structure, composition of these macromolecules like your peptides, polysaccharide, polynucleotide, and nucleic acids. Now come to the next slide. The significance of this molecular diagnostics. To face near futures, the medical practitioner not understand molecular biology, but also imbalance the use of this rapidly expanding body information in his medical practices. So we use this molecular diagnosis to get a medical information about the body, or you can say that the history of particular disease for the particular person, and also it is used to identify that the particular person have a risk of that or not. And this practices can familiar with medicines, pediatrics, oncology, obstetric and gynecology, pathology, or any other medical specialty. Now, molecular diagnostic are transforming medicines. So, first test is what we do a prenatal testing, then disease predisposition, then disease detection, then drug selection and reoccurrence monitoring. So, we use this molecular diagnostics. So, first of all, we have do what prenatal test. After that, when we do a test, we predict that maybe the person have this disease. After that, the disease are detected. Okay, suppose we some person have a low hemoglobin count, then there are many causes of this, so we predict all the causes. Okay, but when we perform a test, then after that the disease are detected, then according to this disease the drugs are selected for the cure. Then when these disease are disappear with the help of particular drug, then there is a we monitor the reoccurrence of that particular disease. So for all these things, we required a molecular testing. Some key questions that asked frequently is that, is the baby healthy? And this is called a prenatal testing. What disease is this patient at a risk? Does the patient have disease history? What drug should I prescribe? And has the disease returned? So this is all about the molecular test. Now, some old techniques of the molecular diagnostic and some new techniques. Like if we talk about the old techniques, then it is totally based on a grow cells or pathogens. Okay, and this have a problem for growing a cell because it takes very, this process is very slow. Okay, and very costly. And this new technique of molecular diagnostic is what? For a direct test. Okay, either what should we use? We use immunological or a DNA RNA based. So this take a like if we compare the time with all the from old and new, then new technique required less time as compared to the old techniques. Now come to the next slide, molecular diagnostics. So when we do a molecular diagnostics, so we say that the system are good detection. Okay, so when we said that system have a good detection, then it will have a three qualities like sensitivity, specificity, and simplicity. If the system has these three qualities, then it will be considered as a good detection system. Okay, now sensitivity means that the test must be able to detect very small amount of target. Suppose this is your target, which is very small, but a test is used to detect this target. This is what? This is your target, and this is your test. Then if it is used, it is able to detect this target, then it will be considered as a sensitivity. Specificity means that the test yield a positive result for the target molecule. When the test have only a positive result, then it will be considered as a specificity. Then simplicity means that the test run efficiently and inexpensively. Okay, when this test is performed on a routine basis, then it will be run efficiently and inexpensively. Now come to the next slide, molecular diagnostics method. This my molecular diagnostic methods, or you can say that immunological diagnostic methods, are divided into a 8 category. The first one is your radioimmunoassay, then enzyme link immunosorbent assay, that is called ELISA, Western blotting, immunoprecipitation, immunofluorescence, flow cytometry, and fluorescence, alternative to antigens and antibody reaction, and immunoelectron microscopy. Now come to the next slide, target antigens and polyclonal versus monoclonal antibodies. The target antigens are divided into 2 categories. One is your polyclonal and another one is your monoclonal antibodies. The monoclonal antibodies are those antibodies who have a one antigen site. Okay, suppose this is antigen site and this is only one, then they will be considered as a monoclonal antibody. This polyclonal antibodies mean that they have multiple antigen site for a target antigen. Okay, suppose this, 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 and this. They have what? They have a multiple site for the um, antigenic site. Now, target for a diagnostic monoclonal antibodies like polypeptide hormones, they are what? They are target for a monoclonal antibodies like chronin, gonadotropin, growth hormone, tumor marker like prostate and specific antigen, cytokines, interleukins 1-8, drug monitoring, cyclosporin, miscellaneous target like vitamin B12, infectious disease like chlamydia, herpes, rubella, hepatitis B, Legionella, HIV. So they are what? They are the target for the diagnostic of the monoclonal antibodies. Now the first one is your radio amino assay. This radio amino assay are very sensitive and it is in vitro assay technique. It is used to measure the particular concentration of antigen. Okay, concentration of what? Concentration of antigens. Okay, and this concentration are detected with the help of antibody. Okay, so what? Like for example, if we have to detect the hormone level in a blood, so we use what? We use this radio amino acid, and they are what? They are very sensitive in vitro assay technique. Now there are some pros, like advantages and disadvantages. Every techniques have their advantages and every techniques have their disadvantage. So this radio amino assay are versatile, means that we are using same principle for the detection of any type, for the detection of any biomolecules. Okay, so same principle should be used. Using the same principle, almost any biomolecule can be assayed. They are very fast. Usually it take only 2 days or less for giving the result. Giving the result. Sensitive, comparable to the most sensitive bioassay, that is less than nanogram per ml. Large capacity, thousands of sample per day, specific antibody dependent. But if it is an advantages, then along with it is also have a disadvantages, like they uses what? Radio activity. So that's why it cause what? It cause a hazard. Okay, they are what? The equipments are very expensive because we are using what? Gamma or beta counter. When we uses this gamma or beta counter, that's why the use of this equipments are very expensive. Okay, so these are what? These are what they are, some advantages and some disadvantages of this radio amino assay. Now the second one is your what? ELISA, that is called a enzyme linked immunosorbent assay, immunological detection. Here what happened? First one is sample is bind to a supporter. Then we added, suppose this is your, see, this is your supporter and we add sample here. After that, we added a bond primary antibody. After that, we wash it. And the body, if the target are present here, then the primary antibody form a bond with it. And the unbound primary body are washed out. Unbound primary body are primary antibody. Sorry, it is antibody. Primary antibody. This primary antibody are washed out. Okay. And secondary antibody, after that we added secondary antibody. Again, after that we wash it, and after that we added a substrate. This substrate are colorless substrate, and if the target are present, then this colorless substrate are changes into a colored product. Now here, same ELISA test method, what should we do? We add a specific antibody which will be bind to a test molecule if it is present. That I already told you. Suppose that this is your target. If this target are present, then when we added a specific antibody that is also called a primary antibody, then it will form a bond with it. And when we wash it, then then it will remove a unbound molecules, and again we add a secondary antibody, and this will form a bond with a primary antibody, and when we wash it, the unbound secondary antibodies are washed out, and when we added colorless substance, if the target are present, then the colorless reaction are then the colorless substrate are changes into a colored substrate, and this will give a positive result, and it is used for a Cauchy high throughput. So this is about the ELISA. See, this is a ELISA variant. ELISA variant, the L spot. Again, this is your ELISA test. Western blot. So Western blot we uses SDS page. SDS page, we all know that it separate according to the molecular weight. Sorry, molecular weight. Okay, and this blot is used the protein in the gels, and they transfer the sheet of nitrocellulose or nylon by passes of an electric current. Immunoreaction, probe with AB and then radio label or enzyme link second antibody. Detection, position is visualized by means of an ELISA reaction. This is the diagram of the Western blot. This is your cathode, this is your anode, this is your separate compound tracking dye, and this if we see this membrane, then this green part is what they are gel, then this outer covering of the green part are what they are transfer membrane, then this pink part are what filter papers, and this blue with white are what they are pads, and they blue are support grid. So this is the diagram of the Western blot, or you can say that this is the setup of the Western blot. Now immunoprecipitation. Immunoprecipitation, what we do, we uses a suitable antibodies is added. This antibodies make a bond to a protein. If the desired protein are present, then protein A or G added to a make antibody protein complexes insoluble. After that we centrifuge it, then we get up pellets. These pellets are what they are antibody protein complex and supernatant, and we discard this supernatant. Okay, so this is about the immunoprecipitation. Now come to the next slide. Again, this is your immunoprecipitation. Immunoprecipitation, sometimes we uses magnetic beads, so that this magnetic bead form to a secondary antibody. Immunofluorescence, we use fluorochromes like fluorescence 490 to 570 nanometer, rhabdomine 515 to 546 nanometer, phycoerythrin. Okay, so these are what they are, immunofluorescence. Come to the next slide. Immunoelectron microscope. This is used for of the structure of a B cells. Lymphoma was stained with two antibodies. First antibodies is against to class second MHS label with 30 nanometer gold particle, and another antibodies are against was first MHS weight 50 nanometer gold particles. Okay, the density of the first one is exceed with the second one. Now this is written, we know how God wrote the book of life. This is the quote, this is a quotation from Bill Clinton. But do we know how to read the book? Okay, we all know that the book are created by God, but do we know that how to read that book? So molecular genetic test. Molecular genetic test is used for analysis of human identification of RNA, DNA, chromosome, protein metabolites. Okay, for their identification. Then it is used to detect the heritable disease related like genotype, phenotype, karyotype, and it is also used for the clinical purposes. Genetic diagnosis. The purpose of the genetic diagnosis is, first one is what? Diagnosis testing, screening, symptomatic testing, parental testing, pre-implantation diagnosis, pharmacogenetic testing, sustainability to environmental aging. Now, genetic alternation. Genetic alternation take place in two. Alternation means that changes, changes. So it takes either by a chromosomal changes or either at a gene level. Pre-implantation diagnosis screening. This prenatal diagnosis or prenatal screening is used to detect the any disease or the condition of the fetus before it's born. Okay, either we see the condition of the fetus or either we see the condition of the embryo before it's born, and also it is used to identify any disease related issues for fetus or a embryo. This is your implantation diagnosis. DNA diagnosis system. How this DNA diagnosis system are work? Bind SS DNA target to a membrane. SS DNA, what they are, a targeted DNA. Then hybridize to level our target DNA or rDNA probe. Then we wash the membrane to remove unbound probe. Then hybrid sequence are formed between the probe and a target molecule, which are detect the positive or negative. Ordering molecular test. So for a molecular test, we don't require any patient preparation. Just avoid the heparin. This because this heparin are interfere with PCR. It stop the reaction. Now we require the specimens, that is fresh whole blood, EDTA citrate, fresh tissue, frozen tissue, paraffin embedded tissue, slides, etc. Specimen handling, DNA based tests, like DNA should be at a room temperature for a 72 hours. Okay. And if we have a RNA based test and we have a specimen RNA, then we use it as soon as possible, around 4 to 6 hours. Then essential information, we also have a clinical information, pedigree if possible, race, reason for testing, inform consist, that is nature of the test, availability of genetic counseling, implication of positive and negative test. Molecular diagnosis. So this is your cystic fibrosis, and this is the example of your sickle cell anemia. DNA fingerprinting in a forensic. So we all know that we are identical around 99.9%, and there is a 0.1% of unique characteristic, and this unique characteristic make a difference into a millions of differences. These differences reside in several places in the genome, often in a microsatellite. Like your polypharmacy include VNTRS, that is your variable number tandem repeat, short tandem repeat, restriction, fragment length polymorphism, and single nucleotide polymorphism. DNA fingerprinting. DNA fingerprinting are introduced by our Dr. Alec Jaffrey in 1985. DNA evidence is admissible in courts. This DNA fingerprinting is used to paternity testing, identification of criminals, immigration disputes, identification of deceased individually with mutilated or decomposed bodies, identify the sperm donor who decorate the Monica Lewinsky blue dress. Now, preparation of a DNA fingerprinting. So first of all, we have to collect the specimens, and this specimens is either collected by our blood, semen, etc. And we have to must take care that because the DNA can easily contaminate it. Other sources like your bacteria, DNA of person collecting sample. And this DNA are also not stable for a long time if it is come in contact with sunlight, heat, or moisture. Now, DNA fingerprinting is a competitive process. DNA from a crime scene is compared with the DNA of suspect. So we have to at least minimum two sample. One is from a crime scene and one is from a suspected. Then after that, we compare this DNA sequences, and after that we take a action. Then we have to extract the DNA. This extraction part of the DNA are covered in our next lecture, how we extract the DNA. There is a different method for the extraction of the DNA. After extraction of the DNA, we do a PCR. PCR is again discussed in detail. We discuss in our next reaction, but it is what it is, a polymerase chain reaction. Here, what should we do? We increase the amount of DNA for better finding. So this is about the PCR. We will discuss in the next lecture. This is first step or what PCR amplification, where this double stranded DNA are denatured and converted into a single strand DNA. Then after that, restriction fragment length polymorphism. Again, this will be discussed in a separate lecture. Finding pattern and all. Some example of DNA fingerprinting, that is your paternity cases, crime scenes. Examples. Okay, so technical consideration, preserve the integrity of DNA sample, avoid DNA contamination and degradation, avoid incomplete digestion if restriction enzyme are used, use standard hybridization condition, use standard PCR primers and procedure. Gene analysis is less reproducible than capillary electrophoresis of PCR products. Test choice are totally depend upon the cost of the test, okay, sample that we required, turnaround time, sensitivity, specificity, positive, negative, predictive value, type of mutation detected, genotyping versus mutation scanning. So this is all about the molecular diagnosis testing or our molecular laboratory. Thanks for the watching. Please like, share, and subscribe our channel. If you have any query, just let us know in a comment box, or you can also send your query to the helpline number that are already mentioned in your slide. You can also follow the ITLS Academy on different platforms like Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn. You can also subscribe our channel in YouTube, uh, and you can also get our app from a Google Play. Thank you so much. If you have any query, just let us know in a comment box.